All right, guys, now let's talk about the Kelvin cycle, okay? The Kelvin cycle is often called light independent reaction or dark reaction, depending on what you're reading and how old it is and who's teaching it. The Kelvin cycle is the second part of photosynthesis. Kelvin cycle actually uses the chemical energy of ATP and NADPH that was created in the light dependent reaction to reduce carbon dioxide to a sugar. Remember, carbon dioxide is considered an inorganic source of carbon, and we need an organic source of carbon to be able to actually, for our cells to be able to use it. So the Kelvin cycle is a cycle, and so what it means is that it's going to regenerate its starting material. In the case of the Kelvin cycle, the starting material is RUBP, or ribulose 1,5 bisphosphate. All right, ribulose 1,5 bisphosphate is actually a 5-carbon molecule, okay? So we're gonna make sure we keep a count of carbons as we go through this cycle. But let's do an overview first before we actually get into the actual details of the cycle. All right, so again, we've already talked about the light reaction, and we know that the reactants of the light reaction are, of course, sunlight and water, and we produce oxygen as a byproduct. We also generate ATP and NADPH, all right? Now, we need more than one ATP and more than one NADPH to drive the Kelvin cycle. In fact, it's going to take 9 ATP and 6 NADPH to create that 1 3-carbon sugar that we actually need, that we get out of the Kelvin cycle. And remember, that's a 3-carbon sugar, so glucose is a 6-carbon sugar, so that means even that has to happen twice. So it's going to take a total of 18 ATP and 12 NADPH to produce 1 molecule of glucose. I want you to keep that in mind, especially when we start talking about cellular respiration and how much we get out of that glucose molecule, okay? So I want you to pay attention to investment versus return when we get there. Now, in the Kelvin cycle, we have carbon dioxide coming in, and we're going to use the energy from ATP and NADPH to fix the carbon dioxide into a sugar. There's 3 stages of the Kelvin cycle. The first one, of course, is carbon fixation. We've talked about that a couple of times. We talk about fixing carbon dioxide into sugar, all right? And so there's an enzyme, a special enzyme found in C3 plants. Now, when we say C3 plants, we're talking about plants that actually produce a 3-carbon sugar in the Kelvin cycle. We also have C4 plants. C4 plants fix carbon dioxide into a 4-carbon sugar first, and then they do the Kelvin cycle. And then you have CAM plants, which actually make acid. It's an acid metabolism. But we'll talk about the C4 and the CAM plants later on. But right now, what we're talking about is what the majority of plants are, which are C3 plants, okay? At the end of the Kelvin cycle, the sugar that you're going to get out of C3 plants will be a glyceraldehyde 3-phosphate. Now, it's a 3-carbon sugar, okay? There's only 1 carbon in carbon dioxide. We cannot take the carbons from RUBP to make this sugar because we have to regenerate RUBP at the end. So what that means is it's going to take 3 carbon dioxide molecules to be fixed to make a glyceraldehyde 3-phosphate. So if it's going to take 3 carbon dioxide molecules, that means I'm also going to need 3 RUBPs, okay? So that means we have 3 5-carbon sugars. Now, the second phase is called reduction. So this is where your NADPH is going to come into play, because remember, NADP plus is a hydrogen or an energy carrier molecule, not a keeper. So it is actually carrying those hydrogens and electrons from the light dependent reaction to the Kelvin cycle so that what the thing that's fixed can be reduced. And then after the reduction, you're going to have regeneration of the RUBP. That's why this is a cycle, okay? So the goal is to start with RUBP, come back to RUBP, and in the process, we're going to drop off a G3P or glyceraldehyde 3-phosphate. So that's the overall goal. So we want to start with this thing, we want to end with this thing, and then we want to get one of those sugars out in the process, okay? Remember, this is a 3-carbon sugar, so we're gonna get 1 3-carbon sugar out of this. Now, so again, process of Kelvin cycle, you have carbon fixation, reduction, and the regeneration of the carbon dioxide acceptor, which is ribulose 1,5-bisphosphate, or RUBP for short. So let's look at it in details, okay? So I'm going to be over here, PowerPoint over there. Hopefully you can see them both. If not, the PowerPoint is in Canvas. So carbon dioxide is going to enter the cycle. So I'm going to have 3 carbon dioxides entering the cycle, but they can only enter 1 at a time, guys. They enter 1 at a time. That's why it takes 3 of these. So 1 carbon dioxide bonds to 1 RUBP, and the enzyme that catalyzes this reaction, very important enzyme, is Rubisco. All right, the enzyme that catalyzes this reaction is Rubisco. Again, remember RUBP is a 5-carbon sugar. There's 1 carbon dioxide. So the compound that you make is temporarily now going to be a 6-carbon compound that is phosphorylated on both ends. Because when we say 1,5-bisphosphate, if you look over here, you see your 5 carbons, 1, 2, 3, 4, 5. On the number 1 carbon and on the number 5 carbon attached to that, you have a phosphate group, okay? So this is where the 1,5 comes from. Bis, they're on opposite ends. So this is a 1,5-bisphosphate compound. Now, so this 6-carbon compound, the carbon dioxide is actually attached to carbon number 2. All right, so it has been fixed into an organic compound at this point. Once it has been fixed to this 6-carbon compound, it's actually going to be broken. All right, so this 6-carbon compound is going to be broken into a 3-carbon compound. So I started off with 3 6-carbon compounds. Now I'm going to end up with 6 3-carbon compounds. And the name of that compound is 3-phosphoglycerate. And yes, you do need to know the names, okay? 3-phosphoglycerate. Now let's look over here. I want you to notice something. When it breaks it, it leaves 1 phosphate group. The phosphates are important here, guys. It leaves 1 phosphate group on each of those compounds, all right? So instead of having a phosphate group on the end of 3, I break those 3 in half, and now I have 6 compounds that only have 1 phosphate group attached to them. So my next step is going to be to phosphorylate the other carbon, all right? So in order to do that, this is where ATP comes into play. ATP is going to undergo hydrolysis, and the hydrolysis of ATP breaks it into ADP, an inorganic phosphate. Remember, we talked about how ATP, when it undergoes hydrolysis, the phosphate group that breaks off will phosphorylate an intermediate, and we call that a phosphorylated intermediate. So since there are 6 of these guys, it's going to take 6 ATP. All right, and I'm going to end up with 6 ADP, and the inorganic phosphate that was once a part of ATP is now going to be a part of that next compound, and that next compound is called 1,3-bisphosphoglycerate. So basically what you went from was a 3-phosphoglycerate. So it's a phosphoglycerate with the phosphate on the number 3 carbon. Then I added a phosphate to the number 1 carbon also. So now it's a 1,3 bis, they're on opposite ends, bisphosphoglycerate. Still a phosphoglycerate, it's just now there's a phosphate group on the number 1 carbon and on the number 3 carbon, okay? So now I have a 1,3-bisphosphoglycerate. This has prepared this compound to be reduced. So now we're ready for reduction, which is phase 2. This 1,3-bisphosphoglycerate, now remember we have 6 of these, right? There's 6 1,3-bisphosphoglycerates. So now we're going to reduce them by using ADPH. All right, so it's going to take 6 of these because I need 1 per compound. So 6 NADPH will become NADP plus. The hydrogens are going to be attached to this molecule, and that that phosphate is going to come off. Because remember, ATP phosphorylates intermediates temporarily. So the phosphate groups that were added here are going to be removed as the reduction occurs. Okay, they're kind of like flags, like placeholders that say, hey, stick the hydrogens over here. This is where you want to put the hydrogens. Reduce me. And so then basically this is changing places with the phosphate that was there. Now that's an oversimplification, but just a way to kind of help you see what's going on. So that's the reduction phase. All right, now when it is reduced, that's when you end up with your 6 glyceraldehyde 3-phosphate groups. But remember, one of those is going to drop out, okay? So one of them drops out of the cycle, leaving you with 5 of these. Now G3P is a 3-carbon compound. So now I have 5 3-carbon compounds. So that gives me a total of 15 carbons. How many carbons did I start off with? 15 carbons. So now these 5 G3Ps can undergo what's called regeneration. All right. Now G3P, glyceraldehyde 3-phosphate, though, only has 1 phosphate group on it. Our RUBP has 2 phosphate groups because it's ribulose bis 1,5-bisphosphate, right? So in addition to rearranging these bonds to make those 5 3-carbon compounds, 3 5-carbon compounds, you're also going to have to put in 3 more ATP. All right? You're going to have to put in 3 more ATP because you have to add in that additional phosphate that's needed to make this molecule be ribulose 1,5-bisphosphate. And once it is regenerated, the cycle goes all over again, okay? So, let's do it again. Because I know that's a little bit confusing. I'm gonna zoom in on it this time, so give me a second to actually get over here and alter the camera. All right. Love having a whiteboard in the room. I'm gonna hate it when Miss Hicks gets this back. Okay, so let's do it again. Kelvin cycle. Carbon dioxide. All right. I'm gonna try to do the carbons. Carbon dioxide is a 1-carbon compound. Carbon's gonna be orange. All right. Carbon, just step on my top. Carbon dioxide has to bond with RUBP, ribulose 1,5-bisphosphate. That's a 5-carbon compound. And it has 2 phosphate groups attached to it. There's a phosphate group on the number 1 carbon and a phosphate group on the number 5 carbon. There's 5 carbons, right? Phosphate groups on the number 1 carbon and on the number 5 carbon. All right, now these two bond together, and this reaction is catalyzed by an enzyme called Rubisco. Must know this enzyme. This enzyme is Rubisco. Now let me point out that at each step there are enzymes that catalyze these reactions happening because this is a metabolic pathway, but I don't need you to know all of the enzymes and all of the substrates, okay? So the blacks are substrates. The enzyme I need you to know in this reaction is Rubisco. This is the only enzyme you have to know for this reaction, for this class. But there are enzymes at each step, okay? So keep that in mind. All right, now, when these two bond together, they form a temporary compound that we don't even bother to name. It has a name, but we're not gonna worry about it. Carbon dioxide carbon is gonna add to the number 2 carbon, and you still have your phosphate groups on each one of those, okay? Now, this first phase is called carbon fixation, okay? So this is carbon fixation. I am fixing the carbon dioxide from inorganic to organic. It's now a source of organic carbon. Now, since this is a cycle, I have to remake this. So I can't use any of these 5 original carbons in my sugar. I'm trying to make a 3 carbon sugar because this is a C3 plant. So that means I'm going to have to do this process 3 different times, okay? So these carbon dioxides will enter one at a time. So if I'm doing this 3 times, that means that I must also have 3 of these. So remember, we're counting carbons as we go around. So that means I'm adding in 3 carbons here. And if I have 3 times 5 right here, okay, so I have 3 times 5 carbons, it gives me a total of 15 carbons. Now, since I did this 3 times, that also means I have 3 of these guys, right? So here I have 3 times 6 carbons, which gives me a total of 18 carbons. I'm going to drop out a 3 carbon compound, so I had to add in 3 carbons to do that. Now, this 6 carbon compound is actually going to be broken in half, okay? So I'm going to go from having a 6 carbon compound, from having 3 6 carbon compounds, to having 6 3 carbon compounds. So now I have a compound that's only made up of 3 carbons with the phosphate group attached to the 3rd one. I broke it in half. So now instead of having 3 6 carbon compounds, now I have 6 3 carbon compounds, okay? So I have 6 times 3 carbons. I still have 18 carbons. Now, there's a phosphate only on one end though. So now I'm going to phosphorylate the other end. This compound, I'm sorry to give you the name of this one. This compound is called 3-phosphoglycerate because the phosphate is on the number 3 carbon. All right, now I am going to phosphorylate the compound by using ATP. All right, so ATP is going to donate the phosphate group to temporarily phosphorylate my intermediate, which is also going to put past that energy, remember? Because The hydrolysis of ATP is a catabolic reaction that is coupled with other reactions to use the energy. So this is going to phosphorylate my 3-phosphoglycerate, which will then give me this compound that has a phosphate on both ends. So now I have 6 of these compounds that have phosphates on both ends because it's going to take 6 ATPs to do this. I need 6 phosphate groups, so it's going to take 6 ATPs. The name of this compound, now that I've added a phosphate group to the number 1 carbon, is 1,3-bis-phosphoglycerate. Okay? Once this intermediate has been phosphorylated, it's now ready to be reduced. And we reduce it using NADPH. So, using the blue, I have 6 of these. I got to reduce all 6. So it's going to take 6 NADPH. And they're going to be turned back into NADP+. They're regenerated, right? So they are going to be used. This right here is going to be used to reduce this compound. So now, what did I do? Okay. This phosphate group right here is going to have to come out. Because remember, it was a phosphorylated intermediate, so that was a temporary phosphorylation. So all 6 of those are going to drop out, all right? Because when ATP undergoes hydrolysis, it's going to end up being ADP and inorganic phosphate. All right, so this drops out. The molecule that I'm left with is glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate is a 3-carbon sugar. The phosphate is on the number 3 carbon, because remember we got rid of the one that we added to the number 1 carbon. So the phosphate is on the number 3 carbon. And I have a total of 6 of these because I've done it 6 times. All right, so this is G3P, or glyceraldehyde 3-phosphate. Now my goal, because all the way through here, I still have 18 carbons. Right here I have 18 carbons. I only need 15 when I end. So now I can actually take one of these out. I can drop one of these carbons out of one of these G3Ps out of here, because in reality, I only need to keep 5 of them. So one G3P drops out to be used by the cell. To either be used immediately or combined to build other sugars, but that G3P drops out, leaving me with 15 carbons. All right, now the problem is I have 5 3-carbon molecules. I have my 15 carbons, but I need them in the form of 3 5-carbon molecules. So they have to undergo regeneration. This was reduction over here. In regeneration, these 5 3-carbon molecules are rearranged into these 3 5-carbon molecules, but the problem is that they only have 1 phosphate for each molecule, so that means I need to use more phosphate. I need more phosphates, so I need to use more ATP. So in order to accomplish this goal of making these 5 3s, I have to put in 3 ATP molecules. And those 3 ATP molecules are used to regenerate RUBP. So the 3 phases of the Kelvin cycle: carbon fixation, reduction, and regeneration. Okay, guys? So if you count your carbons, you can see how that works. All right, that's that on that one. And then next we're going to talk about those C3 and C4 plants.